So DNA. Genotype and phenotype, those are terms that we're going to definitely use next unit. Genotype means those are the letters of your DNA, A, C, G, T, adenine, thymine, guanine, and cytosine. Phenotype is what those letters actually create. So you might have the letters, the sequence A, C, C, G, T, T, and that might actually code for brown hair. Okay, so the phenotype is that physical trait, the visible trait that you actually express. We use DNA to, again, remember it's the operating system. We need it for replication, and we need it to create proteins. It's the base, the template for protein creation. We also use a bunch of technology. Criminal cases, CSI, you see that in Law & Order, you see it in those shows. It's for DNA fingerprinting, looking at the blood to look at missing persons. For 9/11, they actually used DNA fingerprinting to find out, to try to identify some of the bodies from 9/11. Early diagnosis and treatment of diseases. You can do prenatal diagnosis now with DNA and be able to treat prenatally. That means when the woman is pregnant. Improvements in agriculture. They are now able to, for third world countries, use rice, inject different minerals, vitamins, and different nutrients into rice. Rice is very cheap. And so it's very good to be able to do that. It's true, though. Rice is very cheap. It's true. And so it's very good to, so for third world countries where they eat rice all the time, that it's a way to give them nourishment. Understanding how we work, knowing what DNA codes for what genes, where certain mutations might happen, and maybe how to fix it, curing cancers, things like that. And then we can develop medicine, insulin production. We use from pig DNA to create insulin for people who are diabetic, antibiotics, vaccines. So lots and lots of different uses. Okay, good to move on? If not, it'll be online. So, DNA is the genetic. We learned that based on Hershey and Chase. DNA is found in all of our cells except for red blood cells. Why not red blood cells? It's missing nucleus. Doesn't have any nuclei in red blood cells. So if you don't have a nuclei, you're not going to have DNA. It is the physical template. God bless you. It's okay. Physical template for replication. So you create a new DNA. For protein synthesis, you use DNA in order to create proteins. You each have 23 pairs of chromosomes, so 46 total. And each chromosome, and this is on your packet too, each chromosome is one long strand of DNA. So how many long strands of DNA do you have? 23 pairs, so a total of 46. Each chromosome will be a long strand of DNA. So the discovery of DNA. So we talked about that we know Hershey, Chase, and Griffith figured out with genetic material and that genetic material is DNA, but what does actual DNA look like? It was done, many researchers across the entire world were researching at the same time, trying to figure out, it's like a race, trying to figure out what DNA actually, the model looked like. Watson and Crick were the ones that discovered it, Francis Crick and James Watson. In 1953, so 61 years ago, in April I believe it was, that it was a twisted ladder, or actually known as double helix. Please don't put twisted ladder. We say that so that you can see it, you can visualize that it's a twisted ladder or spiral staircase, but it actually, the name of that is a double helix. So write that down if I ever ask you what the shape is of DNA. The thing that I get upset about, not so much too much anymore, but at the time, Rosalind Franklin did not get any credit. Without Rosalind Franklin, she was the one that took pictures of the DNA. Without her pictures of that DNA, Watson and Crick would have never been able to discover the model. The unfortunate part is they waited to give the Nobel Prize until after she passed away, because you cannot give a Nobel Prize to someone who is dead. Rosalind Franklin actually died of cancer that she got from radiation from doing these images. Not just these, but all of her research images. But now we know with all the feminist movement, we now know her contributions. There is a Rosalind Franklin University medical school. There are lots of different schools named after Rosalind Franklin, but she is an important person along with Watson and Crick are still important, but all three of them combined really determined the structure, discovered the structure. So let's talk about the actual structure. We've already sang the song, so we know all this. There's a little more detail in here that I will help you with. Two strands. DNA is double-stranded, so that's important. There are two strands that will make it making it that double helix or spiral staircase. DNA, deoxyribonucleic acid. We love DNA because it is made of nucleotides. Thousands and thousands and hundreds of thousands of nucleotides bonded together by strong covalent bonds. A DNA or a nucleotide consists of phosphate and a base bonded down one side, right? So sugar, specifically deoxyribose sugar, a phosphate, and a nitrogen base. There are 4 nitrogen bases in DNA. Adenine, which pairs with thymine, and guanine that pairs with cytosine. They're split into what we call purines and pyrimidines, 2-ring versus a single ring structure, chemical structure. The way to remember it, there's no easy way, so the way I remember it is pyrimidine has a Y in it, and so the bases that have a Y are pyrimidines. I don't know if that's a good way for you, but cytosine and thymine have a Y, and so they are pyrimidines. Guanine and adenine are purines. You have the asterisk there in front of the sugar and the base because this is what's different between DNA and RNA. Phosphate is the same regardless if it's in a DNA structure or an RNA structure. Deoxyribose sugar is not found in RNA. Ribose sugar is found in RNA. And the nitrogen bases, 3 of the 4 are found in RNA. Guanine, adenine, and cytosine are found in DNA. Thymine is not in RNA. Thymine is only found in DNA. And that is what DNA is made of. Our next slide, I think, is our last slide. That we're going to cover tomorrow. So the top part, the complementarity or complementary, I don't know if you learned that in junior high or middle school. Complementary means that they bond together, always form together. That's in the song, adenine and thymine make a lovely pair. Cytosine without guanine would feel very bare. So guanine and cytosine always pair together, and adenine and thymine always pair together. Another way, if you don't remember the song, you can remember AT, always together. GC, great combination.
Oh, Grace Catherine.
So that's another way of doing it. And who found this out was Chargaff. Chargaff's rule. He was the one who discovered the complementarity of it. And I'm going to give you a question that you will have on a quiz and a test. Ready? Can we stop for a second? If I have 22% adenine, I want to know how much guanine do I have? So think about it. Raise your hand only if you think you can walk me through the process so I can put it up here. If I have 22% adenine, how much guanine do I have? Yes, walk me through.
Okay.
Well, first, what'd you do
Okay,
next?
so there's the same amount of guanine as there is thymine, so that equals 44% total.
Same amount as adenine as there are
Or
thymine?
adenine and
Yeah.
thymine. So that's 44%, and the rest, there's 56% left.
Hold on. So this is 44%. So what did you do next?
There's 56%.
How did you
left.
figure that out?
Well, it's 44 plus 56 is 100.
So you subtracted 44 from 100.
Yeah.
Okay?
And then half of 56, because there's the same amount of guanine as cytosine.
Mm-hmm.
So divided by 2 is 23. Or, not 23. 28.
28.
Yeah.
But you did the exact way that you should do it.
No.
Does that make sense?